Hello, welcome. I'm Dr. Agustín Jiménez. I'm an associate professor in the School of Biological Sciences affiliated with the program of zoology. I earned my bachelor's and master's degree in the Universidad Nacional Autónoma de Mexico in Mexico City and my PhD in the University of Nebraska in Lincoln. I joined the faculty of this institution in 2009, and since then I've been investigating nematodes and other parasitic organisms that move across mammals. The overarching goal of our program is to study the diversification of parasites and their shifts in associations between wild animals and domestic animals in some cases. My primary goal is to understand these changes and the implications that they have for the consolidation of the faunas across the continental divides and the South American and North American continents. To do that, some of my students work on the systematics of nematodes, but they also are versed in the study of the disease ecology of some parasites that infect bobcats and domestic cats, striped skunks and domestic dogs, and other faunistic elements of the North American continent. In that perspective, our approach transitions between the ecological realm, the immediate contemporary changes that we are experiencing in the landscape, and the historical and evolutionary associations that we see in the continent. The ultimate goal of our project, or our projects, is to understand the consolidation of these changes in the diversification of parasites all over the continent. Part of the research that we do implies the collection of organisms, and then these imply parasites. Parasites are going to always be associated with their definitive host, at least during one stage of their development. We are particularly interested in that parasitic stage when they are forming an intimate association with their hosts. The hosts are these organisms that supply the resources for these parasites so they are able to complete their life cycle and their reproductive needs. In this case, we're mainly interested or focused on parasites of mammals. We go to the field and collect a series of these organisms, and then each event is going to be a collecting event. And we are going to inventory and document all of the associations that are occurring in that event. In this case, we have a chipmunk, a wood rat, an eastern wood rat, and then a mole. These organisms are going to be prepared for study as study skins, as the case that you see here, and we will extract their guts. All of the internal organs will be stored in some preservative. In most of the cases, we're going to proceed immediately to do a dissection of these small intestines to identify and encounter and document the parasites that they will have in their guts. The rest of the tissues, we're going to separate and archive for further study. In this manner, we are linking the collecting event of these mammals with their physiology, their DNA, and all of that makeup that will help us explain the association of the parasites with these particular individuals. After that extraction and documentation, we will record all of these events, and we're going to bring them to our lab, where we're going to start inventorying and analyzing their morphology. In most of these cases, these worms will be identified by morphological features that they are going to have that are important for their taxonomic assignation, for the differentiations. And then in some cases, we're going to be able to actually document some of these structures at the very fine detail. These structures at this very fine detail need to be linked with additional information that is going to be part of these worms. And what we're going to do is complete the extractions of DNA of these materials, so we link this specimen, which is the physical manifestation that the life cycle of a parasite is occurring at one place, and at one place in the space, in, let us say, southern Illinois, right? So we document this, we extract the DNA, and then from that, we will do several techniques. And most traditionally, we're going to do PCR barcoding. This implies just simply extract and amplify DNA from the mitochondrial genome of these organisms and sequence it. So we can characterize these organisms and use it to characterize their variability. Once we separate these specimens and then we store them in these special tubes, we're going to separate them and use them for the extraction processes. We're going to archive them and place them in the minus 70 freezer, but we're going to bring them to our station, where we're going to essentially complete PCR with these basic technologies that we're going to have. We traditionally use kits to essentially achieve the objective, and specific primers that will allow us to amplify the genetic material of our worms. This amplification needs to occur in a manner that will make it possible for people to make comparisons. So in that case, we use a set of markers that almost everybody is using, because in that manner, we can make comparisons and achieve a better universal understanding of the systematic relationships between these organisms. For that, basically, the chemistry, the action of basically separating the DNA, reading the frame, and replicating it, it is going to occur in these thermocyclers that essentially are programmed to achieve their objectives through a number of cycles. From those ones, this is my favorite. This is kind of like my battle horse that I reliably use. But it is right now in standby. But that is part of what we do. In this element, we again are synthesizing and linking the diversity that occurs in our planet by linking these sequences with a specimen that is a parasite, but associated that parasite with their host. And by linking all of these organisms and information and depositing them in museums and data repositories, such as GenBank, IDIC-Bio, and Arctos, we can make it universal, accessible to everyone in every corner in the world. Some of the parasites that we work on are internal parasites, intracellular parasites that occur exclusively in some certain organs of these mammals. In those cases, we use modified technology from the PCR to actually amplify and characterize the presence of the DNA of these parasites inside these tissues. For that, we use the technology called reverse transcriptase PCR, or real-time PCR, as some other people call them. And then this allows to see parasites that are intracellular, such as the etiological agent of the bobcat fever, Cytoxan felis, or the black water fever, and several other diseases that actually are able to amplify a small number of copies of the parasite inside these tissues to actually help us detecting their presence, without actually spending the time in doing histological sections that will make the project exhaustive and expensive in terms of time. After the laboratory portion of the lab has been completed, we use the information to model the distribution of these parasites in space and time. We use statistical frameworks as well as statistical and phylogenetic analysis to understand the relationships between these parasites and contrast that against the evolutionary patterns of their mammalian hosts, or these host-affiliated lineages. This is a very important aspect of research that is currently topical. And as a consequence, we invite you to receive this training in this laboratory. I encourage you to join SIU and seek out for additional information that can help you expanding your training on this exciting part of research. So welcome to SIU. We encourage you to apply and join us in the university and in the laboratory. For additional information, please click on the link that we make available for you. Come join us.